Yes, we're going to do it at 9 tomorrow. Yeah. Once you, yeah. I mean, you can wait and see the grade, or you can leave. It doesn't really matter. Okay. This one, Celerity Cells. Celerity Cells. No ribosomes, no metabolism, and so on and so forth. This is simple filterable agent. It goes through the filter that retains bacteria. Range of virus sizes, no numbers. The idea in here, you have to understand whether or not you can see the viruses in the light microscope. Structures that can be found, got it. You can find it. You can find, not any viruses without envelope, naked, phospholipids and protein. This is pretty straightforward, okay? Types of genomes. Understand, please, I want to highlight, understands what does negative and positive mean. Yes. Stranded? Yes. Both double and single-stranded RNA can be either positive or negative. And understand what positive and negative applied to RNA means, right? Other classifications, route of infection or route of transmission. Types of viral capsids, the three that we talked, complex, helical, and icosahedral. Okay? Parts that make up capsid of bacteriophage, tail fibers, tail pins, and the head of the capsid. The head of the phage. That's it. That's all you have to know. Types of genomes can bacteriophage have? Now, I want to be very specific. What we talked about is RNA or DNA. That is it. Usually double-stranded DNA or double-stranded RNA. What do bacteriophages infect? Covered. Do they actually enter the cell? No. It's a genome that enters. Main step of the lytic cycle. You have the picture, know it. Okay? What are the problems associated with bacteriophages as the bacterial control? Resistance, bacteriophage antigenicity, unknown specificity. We don't know if bacteriophage can infect whatever we want to control or not. Lysogenic, same goes for lysogenic. We call such phage temperate. Prophage, you know what it is. Horizontal gene transfer. Transduction. They do transduction. Right? These guys do specialized transduction. The lysogenic phage do specialized transduction. Human virus, main steps. You have the picture. I kind of give you the main steps here. You just have to understand what's going on during each step. Virus attachment to the cell. Receptor. Fusion. Remember that video that I showed you when the envelope fuses to the membrane? That's low pH. What role does low pH in endosome plays in the infection process? It induces fusion for enveloped viruses and capsid disassembly for naked. Where do RNA viruses replicate? Generally, where? Cytoplasm. DNA viruses? Generally, nucleus. Why? Because they need replication machinery. Naked virus exits, cell lyses. Envelope virus exits, cell doesn't lyse immediately. Does that make sense? Am I clear? The receptors for HIV. It's in there. T cells, some folks are CCR5. That's really tiny, small minus. So CCR5 negative. Okay, so understand implications for the HIV infection. I'm asking you. Just it's the question that you saw there. Type of genome, it's RNA, which is positive, but it's reversely transcribed. It is enveloped. RNA is reversely transcribed into double-stranded DNA. Double-stranded DNA is incorporated into the genome. Know the enzymes, reverse transcriptase and integrase. That explains how HIV can be present forever in the CD4 cells in the form of provirus. Integrase, can we get rid? No, we cannot, because we cannot cut out the HIV genome. So far, does that make sense? If I'm going way too fast, it's all getting recorded, by the way. But if I'm getting way too fast, let me know. If you have any questions, let me know. If you have something like, do we have to know that? It's a perfectly fine question. What causes AIDS? Death of CD4 cells. Please understand the implications of CD4 cells death. Macrophages are weak. CD8 cells are not activated. B cells produce little, if any, antibodies because they're not activated by T cells. Mortality. Secondary infections. That's what you gotta know, okay? Most commonly associated. Okay, let me make a closed list, okay? Nothing more than I will tell you now. So this is gonna be for the lungs. Does anyone remember? Carini and TB. For the brain, Cryptococcus neoformans. For the skin, Kaposi sarcoma herpes virus. Okay? And other herpes viruses. Does that make sense? Question? So that's lungs. That's brain. This is skin cancer. Okay. HIV transmission via blood, sexual, vertical. Which two play the major role in the US? Not the vertical. Well, actually, I need to correct. Yeah, well, I should say one, because it's sexual. There is no transmission via blood, and vertical is minimal in the US. Okay? Well, wait a minute. Drugs and sex, okay, which inevitably should be followed by rock and roll. But those two, via blood, what we say is that transfusion doesn't play a role in the HIV transmission in the US. But needle sharing does. People still share needles. Does that make sense? Vertical transmission is few cases here. HART, know what it stands for, highly active antiretroviral therapy. Steps of the virus life cycle. We talked about attachment, reverse transcription, integration, and virus assembly. Okay? Reverse transcription, integration, virus assembly. Prognosis for HIV patient treated with HART. What's prognosis? Is it good? Is it bad? Good. They're going to leave the north. Why do we not have HIV vaccine? Main cause. Yeah, it mutates a lot. Yes, it mutates a lot. Herpes viruses, DNA genome, envelope, well, they large, okay? I don't try to avoid saying the information that's there. Common features, they all DNA virus, double-stranded DNA viruses, they all envelope, they all can establish latency, they all form episomal DNA. I don't remember what we mentioned else, but there were like 2 or 3 things that we mentioned there. No disease and immunocompetent, evolutionary explanation. We are adapted to each other. Does that make sense? Herpes viruses are co-evolved with humans for millions of years. So killing the host is not in their best interest. That's why they do not kill the host. They latent. Alpha neurons. Diseases, well, know that. Transmission routes, bodily fluids, chickenpox, also respiratory. Vaccines, know whether they are available. For herpes viruses, it's easy to remember acyclovir. Vertically, herpes, yes. Possible outcome for the neonate, horrible. That's all you need to know. Severe neonatal infection. CMV, beta, monocytes. Not really any diseases in immunocompetent patients. In immunocompromised, it can cause a variety of diseases from keratitis to hepatitis to pneumonia. Transmission routes. For everything else except varicella zoster, it's bodily fluids. Okay. Vaccine, no. Are there any antiviral drugs? Acyclovir. Vertically transmitted. Possible outcomes for neonate, bad. Does that make sense? They're pretty uniform. EBV and KSHV, gamma herpes viruses. B cells and epithelial cells. Disease associated with each. KSHV, Kaposi sarcoma. EBV, infectious mononucleosis, nasopharyngeal carcinoma, or Burkitt's lymphoma. There's no vaccine. Drugs, we know. Which patients often develop KSHV-caused illness? AIDS patients. Papilloma, DNA, no. Cervical cancer, epithelial cells, vaccine against papilloma. It's kind of it. Polyoma, DNA, no. BK virus, immunocompromised. JC virus, it should be JC, sorry. JC virus, immunocompromised patients. BK virus affects the liver. JC virus causes the progressive multifocal leukoencephalopathy, which PML. It's a brain. Okay. Influenza. Envelope negative strand segmented RNA genome. Strains are classified by hemagglutinin and neuraminidase. Hemagglutinin is used for attachment. Neuraminidase is necessary for virus moving from one cell to another. Transmission route. Respiratory, animal reservoirs, birds and pigs. Why bird flu cannot be normally transmitted between humans? You answered it in a quiz. Treatment options, not many. Vaccine. Understand why vaccine has to be renewed every season. Does that make sense? Because of the mutations. Ebola virus, negative strand RNA enveloped. Symptoms, hemorrhagic fever. That's all I need for the symptoms, hemorrhagic fever. Transmission route, bodily fluids. Zoonotic, yes. Animal reservoirs possible, monkeys and bats. I'm not going to answer these questions. You know this. Okay. Flaviviruses. Single-strand RNA-positive enveloped viruses. 2 flavis causing hemorrhagic fever. Dengue, yellow fever virus. Encephalitis, Japanese encephalitis, West Nile virus. Common between the diseases caused by these viruses, they biphasic. Transmission routes for all of them, arthropods. Actually, for all that we discussed, mosquitoes. Vaccines against yellow fever is approved. West Nile and dengue, not yet fully. Zika virus infection in adults. Rash, retroorbital pain, joint and muscle pain. Pregnant women, it causes microcephaly. Is the large out? Understand why mosquito-borne diseases are not common in the US. Because of the lifestyle. Correct? Remember that, the discussion? Two major types of fungi, molds and yeasts. Molds are multicellular, hyphae are chains of cells if they are septate, and pretty much one large cell if hyphae are non-septate. Mycelium is the bunch of hyphae. Vegetative hyphae used for feeding, reproductive, for reproduction. Yeast, single-cell fungi, replicate by budding. Sexual and asexual spores can be produced by both molds and yeast. Asexual are produced normally, sexual produced when there are unfavorable environment conditions. Pseudohyphae, chains of single yeast cells that stem one from another by budding. Two major types. Conidiospores and arthrospores. Structural differences between them. Conidio and sporangiospores. Sporangiospores are formed in the sporangium and are released from that sphere. Conidiospores are pieces of hyphae that chip off the hyphae. That's all you need to know about those guys. The importance of sexual spores. They formed under the harsh environmental conditions, and they contribute a little to the genetic diversity of fungi. Antifungal therapy. The major concern, eukaryotes, as we are. That's why there's a high level of side effects. Mechanism of action for polyenes, membranes. Agents that they target membranes. Agents that belong to that group, amphotericin B and nystatin. Griseofulvin, inhibition of microtubule synthesis. Flucytosine inhibits the DNA replication. Azoles inhibit ergosterol synthesis. We discussed several, pretty much if the name ends with azole, ketoconazole, fluconazole, clotrimazole, miconazole, inhibit ergosterol synthesis. Mycosis can be cutaneous and systemic. Cutaneous are in the skin. 3 genera of fungi that cause cutaneous mycosis. We talked about epidermophyton, trichophyton, malassezia. Scalp grow and barefoot nails. Tinea barbaris, Tinea pedis, and so on and so forth. Transmission route, either from soil by contact. Mechanism underlining the symptoms. Keratin destruction. Neurotropic fungal infections. We talked about Cryptococcus neoformans, and what was the second one? Not histoplasma. We talked about histoplasma pneumocystis, Coccidioides immitis. Okay, remember that pretty much all of them, Cryptococcus, Coccidioides, Histoplasma capsulatum, and Pneumocystis jirovecii, they all initially transmitted via respiratory route. We inhale spores. Histoplasma and Pneumocystis remain mainly in the lungs. Cryptococcus and Coccidioides, they go from the lungs into the brain, causing the CNS disease. Okay? When I say symptoms, if I ask, the question can be, which of the following fungal agents may cause severe neurotropic disease, meningitis and encephalitis, for instance? And I give you options: Cryptococcus neoformans, Histoplasma capsulatum, or Pneumocystis jirovecii. You pick Cryptococcus neoformans. That's as deep as it goes in terms of the symptoms. Okay? Am I clear? Okay. Specific geographic distribution. For Coccidioides immitis there is. For Cryptococcus neoformans is not. Which cohort of patient is most vulnerable in terms of the fungal infections? Immunocompromised. That's true for all fungal infections. Respiratory, I mentioned Histoplasma and Pneumocystis, transmission from where? From the environment. Geographical pattern for any of them. Histoplasma capsulatum, yes. Any of them dimorphic. Histoplasma is dimorphic. What does that mean? Understand the idea of dimorphic fungus. Well, immunocompromised. Ecto and endoplasm. Ecto is outside, endo is inside. Ecto is involved in the transport. Endo holds all the thank you very much, organelles. Common structural feature for all protozoa, they lack cell wall. Appearance, they flexible. Cyst and trophozoite. Trophozoite is feeding living stage. Cyst is formed when environment is unfavorable. Covered. Method of locomotion. You will have the question related to this. Mastigophora, flagellum, Celiophora, cilia, Sarcodina, Pseudopodia, Apicomplexa, no movement. All epicomplexans are obligate parasites. Remember that and understand why. They cannot move from one food source to another. Okay, that's why they are obligate parasites. African sleeping sickness, Trypanosoma brucei. Now, this is something I want kind of a little digression to calm you down a little bit about. Remember, it's the multiple choice. You will not get the question, what is the causative agent of African sleeping sickness? And you will have to write it down. You will have to pick, so it's easier. Trypanosoma brucei. It's mastigophore, it's flagellated. Transmission route, it's biological vector. That's all you need to know. I'm not asking like tsetse fly. Okay? Actually, let's remember that it's tsetse fly. I don't remember if I ask actual. Yeah, I think I ask whether it's like a mosquito or a fly. Okay? Same will go for yellow fever. If I ask you what transmits yellow fever, you have to know it's a mosquito, not the tick, not the fly. Am I clear? Tissue tropism goes everywhere, from blood to CNS. Symptoms, the person becomes lethargic. Is there vaccine? No. Treatment effective? Yes, but it's not sterilizing treatment. Effective way to control this infection, control the tsetse flies. Naegleria fowleri, primary amoebic meningoencephalitis, PAM. Transmission via the cribriform plate during the Swimming. Tissue tropism. Brain. Is that communicable? Understand why it is not. Prognosis is poor, treatment is not effective, infection is infrequent. Just answer each of them. Cantoamoeba and Balamutia, granulomatous amoebic meningoencephalitis. Transmission via broken skin from the soil. Tissue tropism, well, brain obviously. It's not communicable. Prognosis is different depending on when you start treatment. Often it is very poor. Entamoeba histolytica, amoebiasis, virulence factors, exoenzymes. That's all you have to know. Am I clear? Symptoms: diarrhea, dysentery, colitis. They lead to the intestinal perforation. Transmission: fecal-oral, amoeboma, granuloma that surrounds the amoebic focus of infection. Extraintestinal amoebomas, liver and lungs. Significant health concern worldwide, yes. Preventative measure, sanitation. Malaria. I don't care about species, but I do care about genus, so Plasmodium. Epicomplexa. This is the question that we answered today. I told you to focus on that. Difference in disease symptoms is determined by the differences in life cycles between different species. Transmission by mosquitoes, endemic regions, South America, Africa, Southeast Asia. It's not zoonotic. It's not present in the US because we live inside. There's no transmission between humans. Sporozoite, liver, merozoite, red blood cell, gametocyte, mosquito, zygote, sporozoite. That's kind of a flowchart life cycle. That's the depth that you have to know. Okay, sporozoite is injected into the blood, gets to the liver, becomes merozoite. Merozoites are released from the liver, infect red blood cells. Red blood cells produce gametocytes, sexual. Gametocytes taken by the mosquito. Fusion of gametocytes produces zygote. Zygotes produce sporozoites. The cycle starts over. Okay. Production of gametocytes, here, it's meiosis. Because you produce sexual cells. Gametes, they sexual. Yes, definitive host is the mosquito. Gametes produce zygote in the mosquito. Symptoms: fever, chills, malaise, hepatosplenomegaly. Every time there is an exit of merozoites from the liver, you have symptoms. Vaccine, no. Treatment available, yes. Control measures, vector control. Toxoplasma gondii, apicomplexa, none. In healthy adults, it's pretty much none. Groups: infants and immunocompromised. Pregnant women themselves do not develop the disease, but babies do. Reservoirs for Toxoplasma gondii: rodents, cats, cattle. You can consider cattle to be a reservoir. Humans can understand how humans acquire it. We discussed it in details. Zoonotic, yes. Rodents stop fearing cats. Classification of helminths: cestodes, trematodes, nematodes. it's not 4, I'm sorry, for some reason. It's 3 groups, of course. Okay? Nematodes round, cestodes, segmented tapeworms, and trematodes, flukes, or non-segmented flatworms. Body cavity, hermaphrodites, know that stuff. It's all listed in the table. Antihelmintic drugs, same, they are eukaryotes, we are eukaryotes. Reproduction, blocking reproduction, no, because they don't die that frequently. So we first need to kill the adult tapeworm. Bendazoles, microtubules, okay, synthesis of microtubules, therefore they cannot do cell division, those worms, when mebendazole, thiabendazole is present. Ivermectin targets the gated chloride channel. When channel is open, cells become hyperpolarized and worms are paralyzed. Uncooked pork and beef. I'm talking about taenia. Okay? People consume normally larvae. People shed eggs. Eggs develop into the larvae in the cattle. Okay, what happens to larva? Larva goes from the intestines of the cattle into the muscles. We consume it. Intermediate hosts are pigs and cattle. We are definitive hosts. Squalus, suckers, proglotids. I told you today, intestines, diffusion. Worm feeds itself by diffusing nutrients. Usually asymptomatic. Cysticercosis. Cysticercosis is the migration of larvae from the human gut into the surrounding tissues. If it goes into the muscle, it's myocysticercosis. If it goes into the brain, it's a neurocysticercosis. How can it happen? When people consume eggs, eggs develop into larvae in the human intestine instead of, say, pork intestine. Pathological effects, know about neurocysticercosis. Okay, things like epilepsy, seizures. Steroids. Why we use steroids? Because when larvae, remember I told you, when larvae die as a result of treatment, your immune system starts to attack them. In order to reduce the inflammation going on after attack, we treat patients with steroids. Prevention, sanitation, deworming, checking meat. Schistosoma, fluke, blood fluke. Regions, South America and Africa, Southeast Asia too. Intermediate hosts, snails. Definitive hosts, humans. Intermediate host gets infected by getting the eggs from environment. Definitive host gets infected, remember, schistosoma by swimming in the water. It crawls through the hair follicles. It's not larvae, it's technically Sarcaria. In the human body, schistosoma goes into the blood. And from blood it can get to various tissues, including specifically urinary system. Okay? Remember what happens with urinary schistosomiasis. People with urinary schistosomiasis can develop bladder cancer because it causes chronic cystitis, inflammation of the bladder. Okay? Does that make sense? With blood, infection can go anywhere. There is treatment. How can you prevent? Dry yourself with a towel after swimming. Fasciola hepatica. It's a fluke. Liver fluke. Practically the entire world is endemic. Intermediate host, snail. Definitive host, it can be human, or it can be sheep, or it can be cattle. Intermediate hosts acquire eggs from the environment. Definitive hosts acquire metacercaria by either eating contaminated grass. Okay. Tissue tropism. Bile ducts of the liver and bile ducts that lead to the gallbladder. Food, epithelial cells. Remember we talked gallbladder cancer that arises from the fact that worm stimulates epithelial cells division. Does that make sense? Okay, so it's carcinoma, gallbladder carcinoma. What are the symptoms of fasciolohepatic infestation? Since it goes to the spleen, it's going to be splenomegaly and can actually lead to cirrhosis. Prevention and treatment options. There are treatment options. Drugs like thiabendazole, mebendazole. Prevention, sanitation, cleaning up the plants. And finally, Ascaris lumbricoides. It's a nematode that is common in Africa and South America. Intermediate and definitive hosts, both are humans, because eggs enter, we consume them with a fecally contaminated food, okay? Eggs develop into larvae in the intestine. From intestine they migrate to the lungs. We can draw it, okay? Intestine, that's larva. Lungs, still larva. From lungs they go to the larynx. Larynx / throat, still larva, and then swallowed into the intestine. Adult worm. Develop into adult worm. Okay? Symptoms, think about this. It can be peritonitis, because when larvae crawl out of this intestine, they can take bacteria with them. Pulmonary symptoms, pneumonia and other sorts of pulmonary infections, cough. Okay. Complications, just mentioned. How can we prevent? Stop using human feces as the fertilizer. There's treatments. Generally, why do we use corticosteroids in conjunction with antihelmintic? Helminths do what with the human immune system? Suppress it. When they're dead, they do not suppress it, and we have massive inflammation. We have to use corticosteroids. Okay?